Herbie and I decided that we could have a session on more broader issues that physicists in the past few years have started to get interested in. And he and some of you ran in the past 5-6 years a number of workshops trying to identify new frontiers in biological research that physicists are getting interested in. And one of those topics that we organized 3 or 4 workshops was physics and cancer. What are the opportunities for physicists to help understand, diagnose, and treat cancer? And the other area was aging. So far there have been 3 workshops actually that we organized in one special session that was organized by the American Rheumatological Society last year, which was dedicated to thermodynamics and aging. We believe that these opportunities will open new venues for physicists to enter. Biology is huge, as you know, and our community is much smaller than the biological and biomedical community. But there are very, very, very important questions, and of course, two of them are aging and cancer. So this session will be more of an overview of what currently is understood in these fields and where potentially physics can help. So I will give the. I can continue talking if you want.
I know that.
But yeah, so let's see. In fact, the National Cancer Institute started an initiative 6 years ago of so-called physical science oncology centers. And they funded a number of centers in the United States. This year, 2 got renewed. And so NCI also is moving in that direction, trying to evolve the physical sciences. Phil Sharp wrote about 5-6 years ago a manifesto called The Convergence of Sciences on Biology. Basically, convergence now is the new word of interdisciplinary. The idea is that you converge different things onto something else. And in this case, it is physics, mathematics, engineering, bioinformatics on problems in biology. Convergence actually is not a new term. It was used in the 50s, where people were talking about converging physical mathematical sciences onto sociological and economic problems. But now we're talking about convergence of these sciences of biology. And some of the very important questions against cancer and aging, so we will dedicate a session to these questions.
Okay. Is this on? Okay.
This is Professor Levine from Texas.
So the first thing I'm going to do is answer Dave's question, which is why I'm up here giving another talk. No, and the answer is very simple, because there was somebody who was supposed to give this talk, somebody named Ben Eshel-Jacobs, was somebody you may know from his work over many years on bacterial colonies, and he was my collaborator and our collaborator in Texas for the last numerous years on a variety of problems. Unfortunately, he passed away about a month ago, and so in addition to dedicating this talk to him, because it's mostly his work, I also wanted to say this is why I'm up here giving another talk, is sort of filling in. When you first begin to think about cancer, you come across the most cited paper in, I don't know if it's all science, but it might be in the cancer literature, called the Hallmarks of Cancer. It was written with 2 versions of this paper. And it was an attempt by the cancer biology community to come to grips with an enormous amount of papers, data, ideas, and try to summarize what is cancer. How do we think about the process by which a cell goes from being a normal cell, participating in our life, our tissues in a healthy way, to being growing, eventually, if we don't do something to stop it, eventually metastasizing into tumors throughout the body and then killing the patient. And the methodology they came up with to try to make sense of this was to try to understand what were some of the key properties that cancer cells had to have that would make them behave in the way that then would cause this disease we call cancer. And this is the original version of these hallmarks. And I'm not going to go through all of them, but some of them are obvious. One is that normally cells in our tissues only grow when they get the right signals to grow, and normally they don't get those signals, so they're sitting in our bodies quiescent. but cancer cells somehow have overridden that signal saying not to grow. They grow even when they're not supposed to. So that's an obvious one. There are other ones here that are a little bit less obvious, and it's a useful way to think about working on some aspect of cancer to sort of say which ones of these you're going to focus on. So I'm going to focus on the one that's called tissue invasion and metastases. Tissue invasion and metastases is the fact that normally if you go to your doctor's appointment and discover some type of localized tumor, then of course it gets surgically removed. There's some localized treatment involving radiation or maybe some other approaches. Of course, there are always complications, but you're in good shape. On the other hand, if you go and you're diagnosed with stage 4 metastatic disease, that means that by the time they detect the cancer in your body, the cancer cells have spread throughout the entire body, and then essentially, even after 50 years of extensive effort, it still basically means that there's limited hope, depending, of course, there's many details, many sub-diseases or whatever, but basically, the thing which converts cancer from a problem into a death sentence is the process of metastasis. So that's what we're going to focus on. Here is a cartoon from Weinberg, probably the leading cancer biologist, describing what has to happen as part of this hallmark of tissue invasion metastasis. The idea is, in green here, you see cells that are just sitting there, epithelial cells typically, that are in contact with each other, they're not growing, they're just sort of staying there, and they might be growing, but they're not moving. And then at some point, some of those cells pick up the ability to move, so they leave the primary tumor. They become these cells here that are moving. Those cells then reach the blood system or some other transport system in the body. They travel through other parts of the body, they leave the blood system, and establish secondary tumors somewhere else in the body. So this process is the metastasis is broken down into motility, going into the blood system, which is called internalization, moving through the blood system, leaving the blood system, which is called extravasation, and then germination, which is starting a new tumor somewhere else in the body. And of course, what this cartoon highlights is that in order to go from being in a static situation to being in this mobile situation, the cells have to undergo large changes in their phenotypic behavior. That normally, when we look at our bodies, there are certain cells that are stationary, skin cells, other tissue cells. There are other cells whose job is to move around, like immune cells and fibroblasts and other things that are involved in wound healing. And normally, there's some type of barrier that prevents one cell from becoming this other type, and somehow cancer is undergoing this transition from epithelial cells to these moving cells, which in this community are called mesenchymal-like cells. Cells like fibroblasts and other cells that are supposed to be moving. So cancer cells, the ones that are emerging from our tissues, are not supposed to be moving. The metastatic cascade involves them undergoing a transition to cells that then become motile. So, I'm not going to have time to give you a full talk, but one of the things that we've been trying to understand in asking where can physics make a contribution is understanding this transition from immotile to motile cells. Now, that involves, I would say, a number of different aspects of types of biological physics that we've been hearing about. Because on the one hand, changing from one cell phenotype to another cell phenotype is a transition in some type of genetic network. There's a state, a cell state, a liver cell, or neuron, or cardiac cell, is some cell that has fallen into some attractor, for lack of a better word, of all the genes in that cell being on or off, reflecting the needs of that particular cell. So when you say a cell has undergone a phenotypic transition from a non-motile state to a motile state, what you're really talking about is having some dynamical systems language in which non-motile states are some attractor, and motile states are some other attractor, and you're talking about a transition from one to the other as this dynamical process. So, as in work on E. coli and yeast and other circuits, you can imagine going through the biological literature, which we did, and culling from that what you think are the major contributors to the network governing that transition. Now, it very much helps to have students who like to read biology papers if you're going to do that. And, normally those are not physics students. Physics students, occasionally will read biology papers, but they really much prefer doing calculations. But there are other students out there who really love reading biology students, and this is really a need for an interdisciplinary group approach. You need those people who will sit there and read 100 papers and come back and give you a succinct summary, because there's no way I'm going to ever read those 100 papers. So here's what you get when you try to go through the literature. You find this type of circuit. And I'm not going to describe in detail, but there are a number of interesting things about this circuit vis-a-vis other people who are looking at circuits within the world of physics of living systems. Usually people are doing it in the context of simpler cells. This is obviously a very complex mammalian cell. So the logic is, first of all, you have toggle switches. Toggle switches mean that if you want different states in a system, you need to have a reason why not everything can be on at the same time, or you can have distinct attractors of this network. Well, here you see this inhibition, which means that when this side is high, it will repress this side, and conversely, when this side is high, it will repress that side. So you immediately see that this type of circuit can have multiple different states. And then you can write down equations and solve those states and do everything else, but the topology of the circuit already has to give you some hint as to why you're going to get something that you expect, which is the ability to have these multiple different states. Now, in this network, this side, S, which is called snail, the Z, which is called zeb, names don't matter. When this side is high, you get moving states, multiple states. So these transcription factors control a whole bunch of different proteins that are involved in allowing the state to be motile. This side are high when cells are epithelial-like, and now we already see something which is unusual, although it's not so unusual because we had a very nice talk from Rel's group yesterday that not all regulation in biological systems is done by proteins, by transcription factors. These things, as indicated by the U here, are actually small RNA molecules. So actually the thing which is maintaining the epithelial nature of the states are high values of various small RNA molecules that then prevent the translation of these transcription factors. So there's an interesting twist on the normal circuits, even in bacteria, because we are learning about increasingly small RNAs of various kinds are involved in circuits, but by the time it comes to these phenotypic transitions in mammalian cells, it seems like small RNA molecules are playing a role which is equal importance of transcription factors. So that's one lesson. Now, once you do that, of course, then that's sort of good news for the physics group, because now immediately you say, oh, I need to develop a new class of models that deal with microRNA. I should point out that Rel was working on microRNA back in 2007 with Eshel and myself, and then to develop exactly what types of models are appropriate when the control of networks are microRNAs. Eshel did this Rel did this experimentally in bacterial systems, but helped us think of but was also involved in thinking through what in general would be the logic of small RNA regulation, and then we built upon that to sort of derive this network. So I won't talk about the details. There are lots of details, of course. I'll talk about one consequence you get. And the consequence you get when you go through this is you discover, of course, that there can be multiple states in the system. That's not surprising. Perhaps a little more surprisingly, you discover that that 2 is not a particularly magic number. By the time you're left with the numbers of coupled toggle switches together, and you look at the different states that are allowed in such a system, you can easily find examples where there are more than 2 states allowed. Here is an example. This is as a function of some external element controlling the amount of snail particles. What we see is this is like a sort of a bifurcation diagram. This is a branch that corresponds to epithelial cells, which is low snail, low zeb. Then up here is the branch corresponding to mesenchymal cells, high snail, high zeb. And unlike what you might have naively expected, it turns out that for this class of circuits, it's quite common, and you can quantify by varying parameters and asking for what range of parameters you get 2 states versus 3 versus more. But it's quite common that you actually go through an intermediate state over here, which you can think of as, if this is 1, 0, this is 0, 1, you can think of this as a half. So sort of the different parts of each segment are controlled, are sort of both expressed at medium levels. And it turns out that this idea has been seen, this idea of intermediate states has been seen in numbers of toggle switches and a variety of mammalian circuits. Now, what's the consequence of having cells that are in this region? Well, it's sort of interesting because these cells, you sort of know what to think of, they're epithelial cells. They just stick to each other and they don't go anywhere. These cells are lone moving cells. You know, they just move and they don't stick to each other and they move. So what do these cells do? So it turns out these cells, you know, stick to each other but still move. So how do you stick to each other and still move? Well, you move collectively rather than move individually. You continue with your cell-cell contacts, but you move as some sort of multicellular fluid flow. So to sort of see that, we used a model that we've been developing anyway. This is a simple motility model. I'm not going to even talk about it because I was told I have to keep my talk short. And anyway, we have a variety of models. This is not, many people have done this, a variety of models of cells moving in tissues, and a variety of experiments. I just want to show you what you get by doing a very simple simulation. So this is what's called a wound healing simulation. You have two edges of cells, and at time 0, you let them go and close the wound between those two. And if you have mesenchymal cells, this is what happens. This is a model, obviously. And I don't know how easy it is to see that. Pretty hard, actually. But what you see is, basically, it looks like a gas. You know, you all of a sudden remove the lid, the cells boil off the bulk here, and they start filling up the intermediate region like a gas, because the cells are moving and they're not sticking, so they just move independently. And of course, eventually, you get dense enough region here, and the tissue and the wound heals. What happens if you have these intermediate cells that still stick to each other? It looks very different. Now you have an interface of cells that move and close. The cells don't boil off, but they still move, and they still move while sticking to each other. So the hallmark, if you want to use that term, of this intermediate state are cells that are motile, but are motile collectively. They still stick to each other, they form sheets, they form clusters, and they still are motile. Now, one lesson in this business, or any business that we do with biological physics, but maybe even more so here, is at each stage along the way, you better have some sanity checks. I mean, you can create, you know, great theoretical castles in the air and discover it has no relation to anything. So the good news here is we immediately discovered, again, my persistent graduate students discovered that there was a group across the street that were directly looking at different cell lines, in this case for lung cancer, and trying to classify their motility properties. It was a very funny email because this is a group that used to be in Seattle, and my student sent an email to this person and said, we have this interesting ideas about motility, and we understand you're doing some experiments, and can we compare our simulations to your data? And he wrote back pretty much immediately and said, yes, I think this is very interesting. Your ideas are maybe related to what we're doing experimentally, and by the way, we moved across the street from you. So having a chapter of MD Anderson Cancer Center is a very useful feature of being able to do this work. Lots of biologists, lots of cancer groups get attracted there, either permanently or temporarily. So we tested this idea, or maybe I would say we made sanity checks of this idea by looking at his data of different cell lines, discovering that there were cell lines that seemed to, if you did some large-scale omics consideration, seemed to be in this intermediate state. Those cells did have the type of motility properties that we expected, and most recently, they've gone back and checked at the single cell level, and they've shown directly now, not only at the omic scale, where you mix everything together, but they've shown that in some of their cell lines, each individual cell actually is expressing both proteins. Vimentin is a typical example of the motile state, and ecadherin, which is the typical protein of the epithelial state, and these cells have both of them at the same time. Okay. Now, why is this transition so important? And, you know, so the transition is supposed to be about motility. But then you go and look in the literature, and maybe I'll show the next picture, it's an even better one. And you discover all sorts of papers that seem a priori to be crazy. So you discover papers that, here's a typical one from 2014. By the way, I think we calculated that every year there are 150,000 papers published on cancer. So you can almost, so that's a problem, right? So, okay, so there's lots of stuff out there that you don't know. You have to somehow find some way to get through this vast literature. But here's a typical paper. This is a serious journal, Cancer Research. And so here is the language. Everything dealing with the mesenchymal transition, that's what I'm talking about, somehow promotes escape from T-cell mediated lysis. You might have read, in fact, Science magazine last year declared the breakthrough of the year to be using the immune system to fight cancer. How does the immune system fight cancer? Well, cancer has mutations. It has differences from normal cells. The immune system is supposed to be able to recognize things that are not our cells. So the immune system, in principle, the T cells, are supposed to be able to recognize cancer cells and kill them. But life is not so simple, because first of all, those cells are not that different than our normal cells. It's not like looking for a bacteria or a simple, parasite compared to our own cells with slight numbers of mutations. But also, cancer fights back. So cancer, when it undergoes these transitions, basically creates signals that try to prevent the immune system from attacking them. So this transition not only leads to motility, but it leads to some state which is somehow more resistant to immune systems. It's also more resistant to drug therapy. Now, we think we have some ideas as to why that might be true. It's not at all obvious. So we think that by the time you look at the networks that are not just the one I talked about before, but how it couples to other networks, what you discover is that when the cells become motile, they not only become motile, but they sort of revert back to a more stem-like state. They're sort of more plastic. They're more similar to our embryonic stem cells. It's sort of like going backwards not in evolutionary history, but backwards in developmental history. The cells that are now epithelial cells that are very highly differentiated, part of undergoing this transition also makes them less differentiated, more like our embryonic stem cells, and those cells are actually known to be more drug resistant, less resistant, less sensitive to immune system cells. So the coupling between different networks is equally important to just the pure motility problem. This is something we've also been thinking about. Now, last slide before just making some general comments, because I can talk about this forever. So why is this important? Okay, I mean, it's nice, you can write down models, you can do calculations, you can compare the cell line proteomics data, but it turns out that there's a real possible payoff for this type of thinking that really came up in papers written over the last couple of years in the cancer community, which is, it's well known that metastasis is actually a very low probability event. The idea that a cell is going to leave your liver, or your kidney, or your breast, and make it through the tissue, make it through the blood system, leave the blood system, go to some other part of your body, which is very, very different in terms of its local physiological environment, and start growing there, is actually a very, very low probability event. There's all sorts of reasons why the cell will die, why the cell will get there and not grow, why the cell will be attacked by the immune system as we already. So we know this is a very low probability event. Of course, there are many cells, so it only takes one to actually do this. But it turns out that you can experimentally, in mice, determine, what is the most likely path to get from the primary tumor to a metastatic colony. And what these people showed, indicated by their quantization here of a factor of 50, they showed that the most likely path, the most likely way in which cells can survive this journey is by not doing it on their own, by actually doing it as part of clusters. So they showed that clusters of cells were much more likely to survive buffeting by turbulent flow in the blood system. They showed that clusters of cells were much more likely to be able to get to a foreign environment and at least for some amount of time support each other in this foreign physiology. So they don't have a methodology to explain why this is true. They just measure this in mouse systems. But what we would argue, based on combining this idea with our ideas, is that these intermediate phenotypes that looked like a curiosity. Oh yeah, you can have these cells that move collectively, and that these things somehow have both motility but maintain collectively. They may be the most critical elements in metastasis. And that's the idea we're trying to pursue to see if this idea makes sense. Okay, so I'll quit. There's a lot more to do, of course, always. But I want to talk about final thoughts. There was lots of need for physics here. I don't know if I agree with Tito that 50% of you should work on cancer, but certainly there's lots of room for more people to work on cancer. There are lots of relevant directions to take what I talked about today. If I had another hour, I could tell you about all those relevant directions. If you want to read about some of our work, we have a review article that is published on the archive. I'm still a physicist in that regard, and it will appear in Frontiers in Oncology sometime in the next month or two, I guess, describing what I told you about today. I want to mention one more thing in passing. I'm done with my talk, but this is an opportunity for the subset of you who actually think you might be interested in going in this direction. We're going to have a meeting at Rice. It's a meeting that surrounds a meeting called Hallmarks of Cancer Symposium. This is a symposium, a one-day symposium with some number of leading cancer biologists, with me masquerading as one of them, in there. So this is Bob Weinberg somewhere. This picture, here's Bob Weinberg. This is Arnie Levine. Again, Levine is the most common surname. It's a common thread. And this is some other people. What we're going to do is create an iPoLS event around this. We're going to have a day before where we give some lectures on thinking about physics to fight the cancer. People will then attend this meeting, and then we'll have a one-day discussion afterwards. And if you're interested in doing this, please send me an email over the next couple of days, and we'll try to fit in some number of students from the iPoLS community to be able to come to this workshop. Thank you very much.